This baby, like all of us, started life as a single cell. But by now, he has all the different kinds of cells he'll have as an adult. Even after only 8 weeks' development in the mother's womb, we can already recognize in this fetus the main characteristics of a human being. A hand with fingers, an eye within the newly forming face. How does this specialization happen? What factors influence the process of development? This is perhaps the most exciting and certainly the most fundamental question in all biology. There are clearly obstacles, both ethical and practical, to studying early human development. Amphibians are popular alternatives. A frog lays eggs which develop outside the mother. The embryo can be observed and experimented on at all stages and develop into tadpoles in just 2 days. Not surprisingly then, amphibian development has been the subject of study for over 100 years.
Of all vertebrates, the amphibian is the best described, and I think one could say probably in general embryology, amphibian development is better understood as far as the stages and the events, induction, the specialization of cytoplasm. That's all better described in amphibian eggs than in any other group of organisms.
Speeded up 500 times, here are the early cleavages of an amphibian embryo. 4 cells, 8 cells. Every half hour, the cells divide in perfect synchrony. After 10 hours, there are several thousand cells, all apparently identical.
After this time, when several thousand cells are present in the embryo, then the next really important event in development occurs, and that's gastrulation. The name gastrulation is used because in the course of it, the gastric cavity, the gut, the stomach is formed.
During gastrulation, cells stream inward. A dark line appears, the outline of the blastopore, through which cells move. Inside, they will form the wall of the gut. At the same time, the embryo starts to elongate as cells create a central axis. A trough forms, and the edges of the trough close together to make a tube. This is the neural tube from which the entire nervous system will develop. The top will be the brain, below it the spinal cord. A recognizable tadpole starts to emerge. These tadpoles are the result of normal development. They will grow into normal, healthy frogs. The crucial stage of development is now over. But if that early stage is interfered with by merely chilling a newly fertilized egg, development can go disastrously wrong. Cells which would normally become brain and nervous tissue become gut instead. In severe cases, there isn't even any tail or trunk. Successful development depends on differentiation of cells to form different sorts of tissue. But the great conundrum for geneticists is that all cells within an organism, whatever their specialized function, seem to contain the same set of genes.
The best experiment to demonstrate that cells have the same genes until very advanced stages in development is the one done by John Gurdon on nuclear transplantation.
In the 1960s and 70s, Gurdon conducted a now famous series of experiments. He took fertilized eggs from a normal frog and destroyed their nuclei by irradiation. He then took specialized cells from the intestine of an albino tadpole. He injected their nuclei into the irradiated egg. This egg now had the genes from the intestinal cell of an albino tadpole. If the set of genes was complete, it should be able to direct the egg to produce an embryo that would become an albino tadpole. If the set of genes wasn't complete, normal development would not occur. The experiment was conducted many hundreds of times, and in a few cases it worked, producing identical copies of the original albino tadpole. The generalization from this experiment is that all cells in the tadpole contain the same set of genes. If that's true, then something else must be making the cells different. What we now think makes a nerve cell different from a fibroblast is the number and spectrum of genes switched on. The nuclei of both cell types contain exactly the same complement of genes. In the nerve cell, only a certain set of its genes is expressed. In the fibroblast, some of the expressed genes are different. If this is true, we'd like to know how and when this gene switching begins. To investigate this requires genetic analysis. We need to be able to make mutants which don't develop properly. And this is where we have to leave the frog. It just doesn't breed quickly enough for us to look at mutants. What does is the darling of modern developmental geneticists, the fruit fly Drosophila.
Flies are easy to grow in a laboratory. You can take a male and a female fly and put it in a bottle like this, and after 2 weeks you can end up with 200 to 500 flies. The animal husbandry involved in taking care of flies is much more simpler than virtually any other laboratory organism. Now, as an embryologist, my major interest in flies is in the early steps of fly development, the early stages of embryogenesis. And the reason for this, I think, is that it's in these early stages that the basic body plan of the animal is established. Cells are told or directed to be head, or to be tail, or to be particular segments, or to be mesoderm or endoderm, particular cell types. And what you'd like to know as a developmental biologist is how are cells directed into these particular pathways? Why, what is it? What does this choice that cells make involve turning on particular genes? If so, can we identify those genes? What controls how those genes are turned on?
Here's the developmental pattern of Drosophila. After fertilization, the nucleus within the egg quickly undergoes mitosis. Further nuclear divisions are very rapid. At some stages, a division is occurring every 10 minutes. After about an hour, the nuclei start to migrate to the edge of the embryo. When there are about 6,000 nuclei, they stop dividing and cells start to form. This stage is called the cellular blastoderm. All this has taken place within 3 hours of development. The next thing that happens is that the embryo gastrulates. A fold appears in the underside of the embryo, the so-called ventral furrow. As gastrulation continues, dramatic changes appear in the shape of the embryo. After about 6 or 7 hours, it becomes possible to see the segmentation pattern. By this stage, we know that the segments are committed, some to becoming head, some thorax, and some abdomen. At about 22 hours, the embryo hatches from the egg and starts to feed and grow. It goes through 3 larval molts until it reaches the size of an adult fly. Inside the larva are tiny clusters of cells called imaginal discs, which will grow out and form the structures of the adult fly within the pupal case. Finally, the adult fly leaves its pupal case behind. Just 9 days after it was a fertilized egg, it's ready to lay its own eggs. All this is under the control of just 5,000 genes.
What really should be possible is if there are only 5,000 different genes, it ought to be possible to make mutations in all those genes. If we just make lots and lots of mutations, enough mutations to have hit each gene once, and then kind of sort through those mutations and figure out which mutations alter embryonic development in an interesting way, and then use those mutations as probes for figuring out how genes control embryonic development. And so since it seemed like it was an experiment that we could do and it would work, we went ahead and tried to do it.
Chemical mutagens are routinely used to make Drosophila mutants. Together with a German colleague, Erich Wieschaus made 18,000 mutants to be fairly sure of hitting each of the 5,000 genes at least once. Making and collecting the mutants, though, was just the start of the problem.
What you end up with at the end of a mutagenesis experiment is a lot of stocks. In our case, since we were looking for lethal mutations, we ended up with something like 18,000 different lethal mutations that cause homozygotes to die at some time during their life cycle. Each of those, since those mutations were recessive, each of them could be maintained in a stock where the heterozygotes would continue to breed and produce homozygotes, 1/4 homozygotes each generation. Our job was, though, to find out which of these 18,000 mutations were interesting, that is, which of them had affected genes that were necessary during embryonic development. From a theoretical standpoint, that's fairly simple. All that you have to do is just look at the embryos from each stock, since 1/4 of the embryos are going to be homozygous, and figure out which of the 18,000 stocks are producing abnormal embryos. But technically, 18,000 is a very big number.
They developed a rapid screening process. By using test tubes glued together in blocks, they could handle 19 different stocks of mutant flies at a time. The females will lay their eggs in yeast colonies on the Petri dishes. 1/4 of the embryos will be homozygous for a mutation, so it's a relatively simple matter to observe the colonies for abnormal larvae, or eggs which won't hatch. Surprisingly, only 3% of the mutants showed visibly abnormal embryos, which represents about 140 genes.
If you think about the way an embryonic development occurs, you obviously need more than 140 gene products to make an embryo. You probably, in fact, need most of the genes, products from most of the genes in the entire organism's genome. But the thing that our experiments told us was that most of those substances, certainly most of the boring substances like actin or tubulin or RNA polymerase, which an embryo clearly needs, are going to be supplied maternally, are going to be put in the unfertilized egg, waiting there for the embryo to use.
Intriguingly, some of the most interesting defects in the embryonic development arose not from mutations in the embryo, but from mutations in the mother, so-called maternal effect mutants.
What we're going to watch now is very early development of Drosophila embryos viewed in time-lapse cinematography. Now, this is a normal embryo. What we see here is the cellular blastoderm is just beginning to gastrulate. You can see the ventral side, the cells moving in here. Look at the posterior end here. The cells are making this posterior midgut, and it's being pushed up onto the dorsal side of the embryo. Most of the action is here at the posterior end. If you look at the anterior end, there's not very much going on at all. This posterior midgut moves anteriorly until, after about 30 minutes, it reaches this fold here that's called the head fold. If we look at gastrulation in an abnormal embryo, what we see is that initially the embryo looks pretty normal. It has a ventral furrow, and it's making a posterior midgut at the posterior end, but the important thing is at the anterior end, you also see a second posterior midgut forming. And these cells, which normally have made head, are making posterior midgut. This second posterior midgut moves along the dorsal side of the embryo until the two posterior midguts meet in the middle of the embryo. This embryo will continue developing just as a normal embryo would. The normal embryo would have ended up with, at the end of embryonic development, with a head at the anterior end and a tail at the posterior end, and these segments that you can see easily by the denticle bands and hairs. And the mutant embryo that we've just looked at, which is a bicaudal embryo, ends up with no head, a normal tail at the posterior end, and a second tail at the anterior end. So you have two tails in mirror image symmetry. Now, this bicaudal mutation is one of the most spectacular alterations that you can get in embryonic development by altering genes. One of the most interesting features of this mutation is that the embryo here that we see here is itself genetically normal. It was its mother who failed to put something into the egg that told these cells at the anterior end that they should make head rather than tail.
In the absence of this substance from the mother, an egg will generate its own symmetric pattern. This is a computer model showing the dynamics of this process before the egg is laid. This is the resulting bicaudal mutant. When the mother pumps something into the egg at one end, this process is altered, and the egg generates the normal asymmetric pattern to give a head as well as a tail. So, the geography of the embryo seems to be influenced by signals from the mother.
At some point, though, during embryonic development, the individual cells have to read those signals, have to respond to them, they have to begin setting up their own patterns. And it's in this second step in the process, that we expect the embryo to begin using its own genes.
Here's a normal larva. Mutations in the embryo's own genes lead to various bizarre-looking larvae.
All these different mutations, and there are about 20 different loci that do this, they all alter the segmentation pattern by removing different parts of the normal pattern, and the remaining parts of the pattern develop normally. So you end up with an embryo which has a smaller number of segments. One of the most striking things, though, about these different mutations was that we could easily group them into 3 classes, depending on what the effects of the mutations were. In one class that we call the gap mutations, a big region of the pattern was deleted. So you end up with an embryo with a smaller number of segments because we've left off almost all of the anterior abdomen and thorax. In another class of mutations that are called pair rule mutations, the pattern deletions are very extensive, but they occur in small blocks along the entire anterior-posterior length of the embryo. And the last class also shows pattern deletions, but here they're repeated at single segment intervals, as though these genes are involved in controlling the pattern within the segments themselves.
Wieschaus's lab has become a supplier of segmentation mutants to molecular geneticists throughout the world, eager to study how the segmentation genes operate. He calls it an orphanage. Some were already known before Wieschaus's experiment. This one is a famous mutant called Fushi-Taratsu, or FUTS for short. If we compare it with the normal embryo, it has segments missing. Every second segment, in fact. This leaves a mutant with half the usual number of segments. The simplest explanation is that in the normal embryo, the FUTS gene is expressed at regular intervals in alternate segments. So we might predict that we could find localized regions of gene expression, perhaps even before segments appear. These would mark out regions that fail to develop and so are absent in the FUTS mutant. In the early 1980s, a technique was developed which allowed gene expression to be looked at directly. It was called in situ hybridization. These are normal embryos at the cellular blastoderm stage, treated with a radioactive copy of the FUTS gene. The gene will hybridize to the FUTS messenger RNA. So the regions where the gene is switched on show up as concentrations of black dots.
On some of these embryos, we can see the stripes of RNA localized just around the outer layer of the egg. In fact, they're more easily seen if we change the illumination to dark field, and then the silver grains, instead of being black dots, shine brightly. what you can now see is that only some of the embryos have these 7 stripes. Some of the younger embryos are just showing a uniform distribution of the RNA, and then there's all stages in between where the stripes become more and more distinct. The very oldest of these embryos would just be beginning to show obvious signs of segmentation. So what we're seeing here down the microscope is, if you like, a molecular pre-pattern, which is going to define the visible, the morphological segments, but it's appearing well before there's any sign of that morphological segmentation.
This new technique can be applied to all the segmentation genes and looks set to provide many new insights into their actions. But what about the later stages of development? Once the basic segmental plan of the embryo has been set up, how does one segment become different from another? We have some clues from a remarkable set of mutants in which all or part of one segment becomes transformed into another. This striking mutant Antennapedia has a leg sprouting where normally there would be an antenna. A thoracic structure is replacing a head structure. Another mutant sprouts wings from 2 thoracic segments instead of 1. These abnormal flies arise from mutations in special clusters of genes, the homeotic genes. There's a specific sequence in these homeotic genes called the homeobox, which very closely matches that found in the genes of organisms as different as frogs, mice, and humans. So could this gene sequence be involved in the general control of repeating body parts? And can the humble fruit fly, with its 5,000 genes, tell us anything about the control of development in higher organisms like ourselves, with perhaps 20 times as many genes?
Systems to build patterns must have evolved a long time ago. They must have been one of the earliest features that enabled organisms to become multicellular. And it's not likely that completely different sets of molecules are used in different organisms. So even though we may not look very much like flies, I think that we probably use the same sorts of molecules to build patterns. That's just a theoretical guess, but it's now becoming quite clear that at least some of the genes that are defining different bits of the Drosophila embryo are structurally related to genes that we can find in mammals as well. The conserved piece of the gene structure has been called a homeobox, and it's extraordinarily conserved. Indeed, there are sequences in mammals which you could not distinguish from the sequence in flies simply by chemical techniques. We don't yet know what those sequences do for mammals, but we guess that they are controlling aspects of development in some ways analogous to what we're seeing in flies.
The real job for scientists in the next 10 to 15 years is to figure out how to extrapolate from the detailed knowledge that we have about a model system like flies to the control mechanisms that operate in what's potentially a more complicated organism like a human embryo.